So now Elisa's going to talk about applications of deep learning in medical imaging.
Okay, thank you, Kevin. Well, as well as in other problems in computer vision, there is also a lot of interest in medical imaging using deep networks. So let's say that in this area there are a couple of well-known conferences that deal with medical imaging. One of them is this MICAI conference, and that conference that took place last year in 2015 in October had two tutorials on deep learning and how this could be applied to some problems in medical imaging. And actually there was a workshop devoted to that last year, and next year this year, in 2016, I think it's October, November, there is also the second workshop of deep learning to be applied to medical imaging. Okay, and also this other conference in biomedical imaging also presented a tutorial on this topic. So you have here two references of tutorials on these topics, but medical imaging has a lot of variety of problems that you can deal with. So in that conference, for example, they pointed at some problems that can be addressed with deep learning, like image description or diagnostic by using medical images or signals, like it's been doing this in signal processing. You can do it also now using deep learning. Image reconstruction, image registration, even if it's not here, and some other problems that can be addressed with deep learning. Specifically, there are some applications that has been already done. So here there are also some examples: breast cancer detection, etc., etc. And I'm going now to present you a couple of examples that we are here at UPC interested in. Okay, the first one is detection of malaria parasites in blood samples, and this was a final master thesis of Jaume Fernandez. Actually, Carlos Diaz, who is here, is also working in malaria, but more in crowdsourcing, detecting this malaria parasite, but doing crowdsourcing. But Jaume devoted his master thesis in trying to apply deep learning for this detection. So these are typical images that you have when you have a blood sample. This is a very specific, well, it's a kind of way to take that sample. There are several ways. One is thin and the other is thick, and there are different types of parasites and stages of the parasites. We had the collaboration with Drassana's Tropical Medical Center, that is very well known here in Barcelona, and so through them we obtained the samples to do this work. So these are. Let me tell you that the samples here are inside the red cells of the blood. So they are within the red cells. I mean, they look black, but they are the white ones. And these are like some disturbance that you can find outside the cells that can lead you to bad results when trying to detect what is in fact a parasite. Okay, so one of the problems that we have in many medical imaging problems or application is that we don't have a large data sets. So in our case, we had 38 images. In all of them, there was the parasite in the image, and depending on the cell, you had one parasite and maybe 3, 4, etc., etc. Okay. So the thing is that we had to do some pre-processing before applying a classification technique. So we tried two pre-processing techniques. One was to do some binarization, and the other was to do a previous segmentation. That was not to segment, you obtain the parasite, but to get the cells, the red cells, that you know inside of them you have the parasite. And then we tried two classification techniques. One is based on convolutional neural networks, and the other in support vector machines. For the support vector machines, you need to extract features. Okay. Our work also, well, we looked at the literature, and we found that in that MICAI conference there was a paper to detect cells, not this type of cell, but other cancer cells in breast cancer, and so that was our inspiration to apply these methods. Okay, so let's just say that I'm not going to explain all the algorithms, but let's say that through hue binarization and some display processing, we were like taking patches and creating subimages that these images were the ones to fed the classifiers. Okay, so for example, this would be like two examples where you have the parasite and these two don't have parasite. The resolution is like that, so it's not such a very good resolution, but you know that this is a parasite. Okay, so for the SVM we took some features. We tried many of them. We classified which ones were the important ones. The geometrical ones didn't provide a lot of success, and at the end they were all related to the distribution of the pixel values, and so we got some features, and we applied this to the red, green, blue channel, gray hue saturation value. So at the end we had like 40 features, well, and this is what we fed to a super vector machine algorithm. Okay, so this is the SVM setup, and for the convolutional neural network, like we had a shallow network because we don't have so many instances, so but still we wanted to try. Here you don't need the features, but you feed the network with the sub images, and there is nothing special in this convolutional network. Okay, but still we tried several configurations and we chose one which gave good results. Okay. As I told you, we don't have that much data, so like this hue binarization gave 487 cells. Of those, 105 were parasites and 382 non-parasites. We did a data augmentation, and we used a balanced data set for training. Okay. Well, there is another algorithm which does segmentation but gives worse results, so but we have it here. So these were the results with support vector machines. So don't look at all the results, but let's say that we got like 61% of detection of parasites. And so still the results were not good. We could have worked more on the support vector machines, features and all, but our goal was to see if deep networks work, and in fact we for the hue binarization we got very good results. We failed in only little instances. The cell segmentation didn't have a 100% true positives because the cell segmentation was not good enough. Okay, the part of segmenting the cells. But well, these are promising results. But of course, our database is limited. We need to look for more data to see if these algorithms are good. Okay, so this is one application that we see that at least in this example works better than support vector machine. Okay. Okay, another application of convolutional neural networks for that we are interested in here is brain tumor segmentation. Actually, it's a project that started not very long ago, maybe not even 2 months ago or something like that. And this is also done with Professor Veronica Vilaplana and there are several students involved in this project. Actually, this came from the fact that these students and Veronica are working for the Pascual Maragall Alzheimer Institute Foundation. So that's so they are quite familiar with dealing with information or data coming from MRI imaging of brain. So we said, okay, we know about that, and we also say we want to work on deep learning. So we found that there is a challenge in that conference, which is called BRADS, and it's for brain tumor analysis, that is segmentation. So we are working towards this challenge, and the due date is 21st of July, okay? So, well, I will skip that slide, but just to say you that what is the previous work on this, well, first let me show you some of the images. You have, your data it's in 3D, first of all. Okay, you have volumes and you have different channels, so like different ways of obtaining the data. So for example, here you have one channel, two channels, three channels, and you have the annotated information done by the medical doctors. Okay, so this is an example that you want to detect 4 kinds of tissues in the brain plus the background. So this would be what you ideally want to get. So that's a segmentation problem in 3D, okay? And there is a previous, actually a previous work on that, and it's called Deep Medic. Let's say that the competition last year was won by one CNN proposal. This CNN proposal was done with 2D slices. But these authors, in April 2016, released a new proposal outside the challenge, but using the data that was based on 3D CNN, like Xavier said. Well, now we can deal with 3D convolutions. In this case, it's not time, but it's voxel, it's volume, okay? So, as you will see, when you look at their proposal, they look like any or more typical architectures for CNNs. But the only thing that you have to know here is that instead of having 2D images, you have 3D images, so you have these volumes. You need to do special strategies because the data is complicated and you want a refined segmentation, and you are dealing with 3D, so you need a lot of computation, but you use strategies to save computations, like keeping some of the results of convolutions. To mention that we have to classify every voxel by voxel, which kind of tissues it belongs. And there are 4 classes plus background. So they have this shallow architecture, and let's say that with this group of students, they have reached that stage. So we wanted first, before giving a new proposal, to, let's say, to copy or to reproduce their experiments. So now at UPC we are able to reproduce this shallow architecture and train the network, okay? But they also propose some strategies like you have seen these days with like BGG. So instead of using 5 by 5 by 5 filter sizes, you use 3 by 3 by 3, and you make a deeper network, okay? This is one of the things they did. They also use, like you have seen also in other examples, different resolutions, and at the end you combine the resolution. So now we are in the process of, let's say, you see this architecture and they look like 2D. It's almost transparent that it's 3D, and maybe we're going to use one of the strategies that Amaya has presented, and we have in mind some of them, and maybe if we have time, we explore the one that gives better results. We hope that better than these ones, okay? So, well, here are some results with this Deep Medic. I will skip it. And just to summarize, to say that there is an interest in the area of medical imaging to use also deep learning. You have here a couple of tutorials, and in this conference that I mentioned, and also 2 examples that it is of interest of people here at UPC. Okay. So any questions so far? No? If anybody is interested in this topic, we have some other problems that we are dealing with. Maybe a scholarship for those.